Okay, coming back. Just get started. All right, for the remaining 3 lectures, just a kind of reminder that your course project presentation is 3 weeks away. I know it sounds pretty fast, but yeah, just look at the calendar, and in 3 weeks, you're expected to do the course presentation. All right. So for the remaining 3 lectures, and this is the first one that we will talk about the ChIPseq experiment. First, just as usual, we will review a little bit what we have talked about. And in the let me adjust this. In the previous lecture, we talked about alternative splicing, and just a little bit review. And you can see this is one gene that has 3 different exons. And in one condition, all the exons will be in the final isoform. In another condition, the middle one has been skipped. And this process is called alternative splicing, which is a process where one gene produces more than one type of mRNA. And the technology measured alternative splicing will include the exon array, exon junction array, and RNA sequencing. So just a little bit reminder for the exon array, that is actually you design your microarray using each individual exon as a unit rather than using the entire gene as a unit. And the exon junction array is the platform that you design your probes to target on the junction side. So if you still remember that slide. And of course, the purpose of this course is to talk about RNA sequencing experiment. And in terms of using RNA sequencing to study alternative splicing, and there's 2 general approaches, 2 categories. The first one is the so-called isoform-based approach. So you can see that this we will look at this picture in this direction. So this is one isoform, another isoform. Sometimes you can have many different types of isoforms. It can be, for some extreme cases, you can have hundreds of different isoforms in one cell. And the purpose of this type of analysis is based on the RNA sequencing measurement, we want to reconstruct the different isoforms and also trying to quantify the expression levels of different isoforms. And another direction that this goes is event-based, alternative splicing event-based approach. So instead of trying to figure out the different isoforms and their expression levels, and I would rather to look for each individual event. Okay, I don't care whether it's in which isoform or how many isoforms it's in. And eventually, my measurement will be for this particular alternative splicing event, what's the percentage of final gene product, have it or not having it. So this is called the percentage of spliced in, if you still remember. And MESO, which is a mixture of isoforms, was designed to focus on this issue. For the isoform-based approach, that we have genome-guided reconstruction approach and genome-independent reconstruction approach. And basically, this is a alignment-based. So you first need to do the alignment, but for the independent reconstruction is something you have to do the assembly first. Okay. All right. And one of the major things that we need to go through, and I do expect that you understand completely, is the differences between the Cufflinks and scripture. These 2 tools that we talked about last time, and both of them are for the genome reconstruction and quantification purpose. I want to first mention the commonality of these 2 tools. And the first one is both of them are genome-guided reconstruction approach, so you have to do the alignment first. And the second one is both of them are heavily rely on spliced alignment. So it has a special requirement for the alignment quality. And most people just use the bow tie, and I think that is very appropriate. Of course, you can also have some other aligners, but they have to fit into the RNA sequencing category so that you can have the spliced alignment, the rates that across junctions, and both of them heavily rely on that. And another thing that for these 2 algorithms, the basic differences is for the cufflinks, it's trying to minimize the path and maximizing the specificity, meaning that maybe I'm missing something here, but if I say it's there, and it's better to be there. So they want to really maximize the specificity of the isoform identification. Well, for the scripture is maybe I say something there is not there, that's okay, but I don't want to miss anything. So they are trying to maximizing the sensitivity for this. This is the major difference between two. And probably at the current stage, cufflinks are used much more than scripture. And one of the major reasons for that is the cufflinks can give you the quantification as well. So not only gives you the reconstruction of the isoforms, but also estimating how many what's the expression levels of different isoforms. And there are some other follow-up tools that follow this cufflinks, including the cuffdiff. So basically, you can look for the differential alternative splicing and to that regard. And we also mentioned the MESO, which is the alternative splicing event-based approach. So basically, what they are using is actually inclusion rates and the exclusion rates. So you can see some of the rates, they support these inclusion events, and some of the other rates, they support these exclusion events. And MESO actually very creatively use the constitutive rates. So basically, they use the Bayesian approach to assign these rates back to different the inclusion and the exclusion events. And from this figure, that you can see that both of these two approaches are kind of are pretty accurate, but by using the constitutive rates, which is the yellow part of it, and your power of analysis will be, so you increase your statistical power so that your estimation for that is you have narrow confidence interval, which is more accurate. Okay? And but the key point of MESO I want you to remember is alternative splicing event-based approach rather than isoform-based approach. And after talking about alternative splicing, we also mentioned that using next-generation sequencing to study gene fusion. This is another major area of RNA sequencing experiment. So a gene fusion, a fused gene is a hybridized gene by joining parts from two previous separated genes at the transcript or genomic levels. So basically, there is a gene, two genes, they don't supposed to be together. They're separate apart very far away, or sometimes in different chromosomes. But for some reason, in a genomic level, they are connecting with each other. So when you do the transcription, and they are fused together. And these sometimes we have observed that a lot of recurrent gene fusion events in different types of cancers. So that people are really very much interested in that. And using next-generation sequencing to study that is, the first one is you can use the pair-end rays to study gene fusion events. And it's like this. So this is one gene, this is another gene. They don't supposed to be each other, but in the reference genome, but somehow they fuse together. And then once you do the pair-end rays sequencing, so one pair, one end are aligned to this part, and another end aligned to this part. Okay, and keep in mind that they don't supposed to be together in the reference genome. So this is called the discordant rays, discordant rate pairs, and you don't know where the based on this discordant rays, you are trying to figure out whether gene fusion has occurred. And the problem of this pair-end rays, only based on pair-end rays and this discordance, are you probably will be pretty confident that the gene fusion occurred, but exactly where the gene fusion occurred, you probably don't know. You cannot go to find out the break points. And that's why that some other strategies is trying to use single-end rays or a pair-end rays on those ones the fragments that is on the breaking point. So for this one, the power of the analysis is become much more difficult. So probably the power is not that high. But once you find something, you identify the exact genomic location where the fusion occurs. And that is very important. And in terms of the algorithm discovery, that we know that regular alignment will miss the fusion genes because those fusion genes, they are very far away from each other. Sometimes they are even in different chromosomes, so you don't really identify those. Most of the algorithm that trying to study gene fusion from RNA sequencing data is on the algorithm development part. So basically, you want to design or apply novel algorithm to do the alignment, just a specific target on the gene fusion. The first strategy people are using is a de novo assemble-based approach, which Trinity or trans-Abbas, so we talked about Trinity last time already. And those can be really important. And also, there's some other algorithms that's based on pair-end-based approaches or improved alignment-based. This is a single-end-based approach. And I'm not going to go details of each individual methodologies, but as you can see from here, that most of the publications were published in this year. So this is a still very fast evolving area. And we also talk about allele-specific expression. So allele-specific approach can eliminate environmental or trans-acting factor. So we mentioned that gene expression actually is affected by the cis-acting factors, which is written on the DNA, and trans-acting factors, and also environmental factors. And if we do the allele-specific expression, we study that, and you can really rule out the trans-acting factors and the environmental factors, only focus on the cis-acting events. And because the two alleles inside the cell, they serve as a within sample control, and they control for each other. So there are different technologies to study allele-specific expression. The first one we talked about is called the padlock, following the sequencing assay. And this is developed in George Church's lab at Harvard. And it's very specific, and the performance is very nice. And of course, for this course, we're talking about RNA sequencing experiment. And we want to make sure that all alignment methods are balanced. So these are the potential limitations. And all alignment algorithms are balanced towards the reference allele. So when you want to see the differences between two different alleles, sometimes that can create additional bias. And also, the RNA sequencing requires both sequencing the DNA and RNA, and sometimes it's too much resource consuming. Most of the groups, they don't have the resource to do this. And also, it requires very deep sequencing as well. So hundreds of reads per base pair may be required for subtle variants, meaning that two allele, their different expression differences are 1.5-fold or less than that. And I guess you can still study allele-specific expression at this stage, but don't really count on those. Once you propose something, if your focus is trying to study allele-specific expression, don't overstate your goal. Sometimes you won't reach it, and if you're not careful enough. And especially if one reviewer knows about that, it's never a good sign. And we also talk about RNA editing, and so RNA editing does exist, and there's a canonical editing pathways. It's basically from A to G or C to T, and both of them are de-animation. So you can see from this example that they have to sequence both the DNA and the RNA elements, and then compare the differences between their sequences. And I presented one study published in Science earlier this year, and but later there's several articles from Nature News that challenge that study. So careful bioinformatics is required to study RNA editing using the RNA sequencing experiment. And the last topic we talked about last time was the microRNA sequencing project. So you can see this is called a small RNA sequencing experiment. So basically, briefly, so what they do is you got a very short sequence. It's about 22 base pair or around that range. You want to put 3' linker and 5' linkers there, and then you want to sequence it. Okay? And so there's several different steps. Usually that for the small RNA sequencing experiment, we use the 35 base pair sequencing reads. because there's really no points to use longer ones. And even for the 35 base pair arrays, sometimes we are overkilling, right? Because the small RNAs, they are only 22 base pair. So we know that the sequence will occur from here, and after the microRNA, actually it goes into certain part of 3' linkers, adapters already. And we talk about two different tools. The first one is DSAP, and the deep sequencing small RNA analysis pipeline. There's a couple of steps. The first one is to remove the adapters, because we know that we are sequencing 35 base pair, but those small RNAs are only 22 base pair. So we are sequencing into the 3' adapters. In the analysis part, we really have to get rid of those. And this is to remove the adapters. And then the second step is to cluster the reads into unique sequencing tags, because you really get so many different reads, and a lot of them are from the PCR artifacts. And they basically want to get rid of those. They just unique the reads they eventually get and continue with the follow-up analysis. And the next two steps are look for the existing database. The step number 3 is match the sequence you find with non-coding RNA database. And the last one is to match with the known microRNA database in mirror-base. Okay? As you can see, very straightforwardly, this procedure. But the problem is you can only study known microRNAs using this procedure. What's going on if we have some novel microRNAs? Because we know that we have plenty of novel microRNAs that we want to study as well. And this paper, which is published in Nature Biotech in 2008, proposed a method to do the de novo microRNA identification. So before we have this paper or this type of study, it's kind of, if you think about it, it's kind of straightforward, right? You got the RNA, microRNA, you remove the linkers, and then you map it against the genome. So if there's a region that is already mapped to the existing database, great. If not, next step you want to take a look at, probably you want to search for the local sequence area to look for those hairpin loops, right? And if they happens to have pretty strong signals, and also it forms local hairpin loops, you probably are pretty confident that there's a microRNA there. But this paper really refined that approach because they are using the biochemistry strategy that microRNA genesis, biogenesis, so what the process is. So basically, you got the microRNA precursors. They will form this type of hairpin loop. But we know that dicer cleavage will only occur at this part. So if this really happens, if this is a real microRNA, you are looking for this type of sequencing features when you align it back to the genome, right? And so this is really because of the dicer really cut this hairpin loop from this part. Sometimes if this is non-microRNA event, the cutting can happen any place. So you may see some other features, patterns in terms of the read distribution along this area. And so by this way, it gives you a better idea whether this is more likely to be a microRNA or not. So you are using the sequencing reads, using the alignment, and using the hairpin loop. So both these two were mentioned earlier. But now you are using additional information, which is the dicer digestion and how the pattern you're supposed to look at. And this will give you a better specificity when you identify the variants, identify the novel microRNAs. OK. Any questions regarding the previous lecture? No? All right, good. So let's move on. Today we're talking about ChIPseq. So I will first give a very brief overview of ChIPseq experiment. And then, as usual, I'm going to present several applications using ChIPseq experiment. All right. You'll be surprised that people are pretty creative in terms of using this methodology that has been well established. And also, in the last couple of slides, I'm going to talk about experimental considerations. Okay? Coming back to this slide that we have presented earlier, and this is the central dogma of molecular biology. You get the DNA and transcription, and they generate the precursor microRNA. And then this precursor will become a mature microRNA, and this is the RNA processing part. We also call this the post-transcriptional regulation. And from mRNA, and it will be translated to protein. So central dogma, pretty simple. And for the ChIP-seq experiment, what the goal here is trying to study this part, the transcriptional regulation part, which also includes two different components. One is the epigenetic regulation, and which include histone modification, DNA methylation, and a few others, and also the transcription factor binding. And we know that ChIP-seq is perfect for study this level. Okay? And but if you really modify the protocol a little bit, and the ChIP-seq or some other type of immunoprecipitation-based sequencing assay, and we can also use that to study alternative splicing and the microRNA, which I'm going to give you a little bit example today. So, but overall, that ChIP-seq, traditionally, what we are referring to is to study this level and to study the histone markers and the transcription factor binding. This is almost the same information as the previous slides. So this I just thought this is a beautiful figure from a user's guide to the ENCODE project published this year in PLOS Biology. I really encourage you to read this paper, and especially if you want to work on this sequencing data and without having too many in your hand. And this has most of the information you need to know regarding where to get those information. But the point here is we know this is our chromosome, and it's very tightly packed. Okay? If you zoom in here, you open here, you will see that this is nucleosomes, and these are the histones. And our DNA actually is wrapping around the histones, right? And for this part, you can see that the histone is really, the DNA is wrapping around it. But some of part, the histone is gone, and you can see that it's pretty much open. And this part is the so-called DNA hypersensitivity site. And then after that, and you can see that this is RNA polymerase, and transcription occurs, and transcription factors most likely binds on that. Okay? And in terms of a ChIP-seq experiment, and the goal is really to understand all different levels of chromosome arrangement during the course of epigenetic and transcription regulations. So there's different levels of cis-actin regulation. The so-called cis-actin regulation are the regulation occurred in the DNA level. Right? So the first level is actually the epigenetic regulation. It's nothing to do with the transcription factors, and which includes several components like chromatin occupancy and chromosome looping, histone modification, and DNA methylation. Those are the epigenetic effects that is written on the DNA code, and at the same time will affect the transcription. And we call it epigenetic regulation. And there's another level, which is the transcriptional regulation, and this will tell you the transcription factors, which are those proteins binding in the promoter region of the gene and affect the gene expression. And also the enhancer binding proteins. And there's some proteins binds in the very much distal location comparing to the gene they are targeting. And those are a little bit harder to study. And also some of them are working on the mediator proteins, which we'll get into a little bit in the future slides. Okay? So the ChIP-seq experiment can help us to study the epigenetic regulation and transcriptional regulation. And also if you are studying, if your ChIP-seq, you're targeting the RNA polymerase II binding proteins, and this you can use that to study the transcriptional activity. Okay, so before the next generation sequencing technology, people still have other ways to study the protein-DNA interaction, and this is called the ChIP-seq experiment. And I think there's not too many ChIP paper published because it was invented, I think the first paper is 2005, 2004, or something like that. And it's very expensive. It's a hard experiment to do. And then shortly after that, the sequencing platform comes out. So this is kind of a short-lived technology, but still gives us a lot of biological insights. So the ChIP-seq is a genome-wide identification for binding sites of DNA or RNA binding proteins. Okay, the same thing with the ChIP-seq. So what goes through here is you got this double-strand DNA, okay? You got a protein binding on it. And your goal is, this is a POI, stands for the protein of interest. And your goal is to identify in the genome-wide where this protein binds on. And the experiment to do is the first stage you want to do the so-called cross-linking. The cross-linking meaning that if this protein binds on the DNA, make it keep binds there. Okay, don't separate. So you just keep binding there. And then at this stage, you want to share the DNA, meaning you want to break the DNA into smaller pieces. And next step, you want to do the immune precipitation, meaning that you can use antibody or other type of affinity-based assay to capture the protein you want to study. Okay? So it's trapped here and captured in this matrix, but everything else will be floating around. So at this stage, if you do the purify, amplifying, and the labeling, and basically at this stage, you can throw out everything else because they are not on the columns you designed. And then you got this small DNA molecules. They originally binds on the protein of interest. And then you put those on the microarray experiment, on the microarray platforms, and trying to see which part of genome has been lightened up. And in terms of the array part, the design generally has two different types. The first one is the so-called promoter arrays. Okay? So what happens is you got the gene, and this is the upstream a couple of thousand base pairs. And you design probes to target on the this annotated promoters or CPG islands. Okay? And there's a couple of platforms you can do that, and there's Affymetrix, Nimbogen, Agilent. They all have their own platforms in terms of design these promoter arrays. Okay. And there's another more expensive approach, which is less biased. It's to use called the genome-wide tiling array. Okay. For this one, the difference between the promoter array and the tiling array is, for the tiling array, I don't really care about the annotation of the gene. Yeah, there's probably a gene here. I don't care. Okay, as long as it's the non-repetitive region of the human genome, I'm going to design my probes to target on that. And as you can see, there will be a lot of information related to that. And in the Affymetrix platform, what they did is they designed this 25 base pair probe. Okay, 25 base pair. And they will put about 10 base pair linker there. So every 35 base pair, they will have a probe there. And they target the entire human genome, which are not repetitive regions. So as long as it's non-repetitive regions, they will target. So as you can see, there's a lot of information there. And for one set of human genome tiling array, even for the Affymetrix platform, you do need 7 arrays to target on the entire human genome. So that's a lot of resource to go through. For the bioinformatics part, and you got the data from the chip, and then somehow you figure out the signals and which part of genome is up or which part of genome is down, the signals high and low, and then you are trying to infer which part of the protein originally binds on. Okay? Any questions at this moment? No? Everybody are crystal clear, or everybody are just confused? I hope it's the clear part. All right, now I'm going to tell you one study. Actually, it's from Indiana University here. It's a collaboration with Dr. Haru Nakashachi in the Cancer Center. And the goal here is to study the effect of AKT, which is a signal molecule, on the estrogen receptor binding to the genome. This is in the breast cancer MCF7 cells. And the array platform that we used was Affymetrix human whole genome tiling array. As you can see, this one is array samples non-repetitive genome with 35 base pair resolution. So every 35 base pair, they will put a 25 base pair probe to target on those regions. This contains 7 arrays with 42 million probes in total. At that time, we were pretty scared, and there's a lot of information to go through. And So we can use this data, they generate this data, and we want to answer certain type of questions. The first question that we are interested in to look for is estrogen receptor binding genome-wide landscape. So you can see there's 4 different lanes here. And the first one is MCF7 in a control condition, meaning there's no estrogen treatment. And you can see there, we did identify some of the regions that are potentially binding site of estrogen receptor, but it's really not too much. And after estradiol treatment, and you can see that the estrogen receptors start to bind everywhere in the genome. This is the entire chromosome 1. I think so, yes. The entire chromosome 1. So you can see before the treatment, it's like this. And after it, and it starts to bind every place. And the same thing happens with the AKT treated and after the estrogen treatment. So there's some basic summary statistics that we found that there's about 4,000 something new binding site comes out after estrogen treatment, and there's some overlapping, and I'm not going to go to the details. But this is the first question, the genome-wide binding patterns of estrogen receptor. And the second question here is the relative locations of ER binding sites with known genes. Are they binding pretty close to the known genes or far away from it? So we did look at our data, and we want to see that you can see the red part is a kind of a random case, but clearly that with our data, and we see that estrogen receptor is more binding preferred towards the close to the transcription starting site of the gene. Although there are still many of them that are far away from the transcription factor, transcription starting site, but in general, they are more enriched close to the gene area. And at this stage, we also are interested in, for the regions that we identified, are they evolutionarily conserved? So you can see pretty clear that they have a very significant conservation evolutionarily. So that really tells you that estrogen receptor binding sites are something useful that even across other species. And the next question that we want to know is there any other cofactors related to estrogen receptor binding? So some other transcription factors work with our estrogen receptor and to do something together or not. And then, so from very basic motif finding algorithm that we were able to find several other cofactors of these transcription factors, and they are cofactors of estrogen receptor alpha. And in terms of this part, I'm going to introduce the methodology, very detailed methodology in the next lecture, and how do we identify the enriched motifs based on the data, ChIPseq data we got. And we also wanted to look for, okay, there's a binding of estrogen receptor, but will that have consequence, meaning of their target genes or the target gene expression level has been changed. And also, of course, we see the correlation of AKT-induced changes in ER alpha binding with E2 regulated gene expression. And we found those numbers of genes that have the correlation. And once we run through the ingenuity pathway analysis, and you probably cannot see from the back, but you can read from the handout. And this is the E2, and this is the AKT. So the genes that are differentially expressed are scattered around. But the two most important factors that we are studying here are something in the central. This is really tells us the data is biological significant, and it's really produced something that we expect. Of course, we want to learn more from that as well. So this work was based on the ChIP experiment. The reason I'm talking about this in such great detail for obsolete technology is most of the things that I'm doing here and we got from here, we can still do the same thing with the ChIPseq derived data. Okay? All the technologies are still valid and our algorithms are still valid. And some of them we will go through in a little bit more detail in the next lecture. All right, let's go to the ChIPseq part. So the ChIPseq stands for chromatin immunoprecipitation, follow next generation sequencing. And the goal here is to identify genome-wide binding patterns of a protein interest. And this protein can be a transcription factor, can be a histone mark, and can be RNA polymerase. It depends on any DNA binding protein, you can study that. Actually, I'm going to get into that a little bit later, that with modified approach that you can also study the RA binding protein binding pattern as well. Exactly the same figure, not going to go through again. And the only difference is here, after you got this fragment that originally binds in the protein of interest, and you do not do microarray, rather you go ahead to sequence them, and then we should be able to find the signals that we want. This is exactly the same message, that's just a different figure, and published in Nature Review Genetics. And the reason I put here is it's pretty, and it really gives you the idea of what we are doing. So you can see that this is a histone, and the DNA is wrapping around it. And there are different histone modifications. So you can see the different modifications. And also, there's the transcription factors that bind in those regions, so other DNA binding proteins. So the point of this part is once you design the antibody, you can either target on this transcription factor or DNA binding protein, or you can target on this histone mark. And then you get the fragments, and then you sequence it. This is, so you can see this is figure. It's very much like a bridge amplification. This has to be the Illumina technology, and these are the beads. Or remember, keep in mind, this is the emotion PCR, and so you can see the beads technology. So it goes to either full 5 or a solid. And this one, you don't need to do any amplification. It directly goes to the helicose to do the sequencing, although helicose is not with us anymore. Right? But so the point here is this is the ChIPseq assay. There's a major steps. The first one is the cross-linking, and the cross-linking, what you do, you use is formaldehyde. You use formaldehyde to treat the DNA, and that will introduce the cross-linking. And but I want to say one more word about this is formaldehyde introduces the protein-DNA interaction, but at the same time, introduce the protein-protein interaction. So if you got a protein complex, right, if one of the proteins binds in the DNA, and you target any protein in this protein complex, that part of DNA will be grabbed down. So this is something that you need to keep in mind. And from next lecture, that you'll know that sometimes we want to use this type of feature to help us understand some of the mechanisms. And the second step is to do the sharing. This sharing can be the sonication, break it physically, or you can use enzymes to treat the DNA. And this part we will talk about in the later slides, that this is one of the major considerations when you do the experiment. And the next step is the immunoprecipitation. Of course, you use antibody to target it, and then further do the sequencing. So these are the major steps of the ChIPseq experiment. Okay, yeah. Where is the question? Okay, there, yeah. We can, so once we go to the different studies, you will read that more carefully, and usually it's about 200 base pair to 300 base pair. You only sequence the beginning part of it, right? So remember, it got fragments, short reads, right? We only sequence the very beginning part of it, and in the middle we won't. But if your break, if this cutting is random, which you use sonication, and that, in theory, that generates more random cutting locations. And if this is a DNA, you can cut here, sometimes cut here, so you have a better coverage of the local fragments. We will talk about that next time. There are some special technologies to look into that, because one of the things that people are taking advantage of is the directionality of sequence reads. And that will help us to narrow it down. We'll talk about that next time. And you won't get one base pair resolution level. So you wouldn't know exact location of those DNA binding locations. And at that time, that also we'll talk about that next time, is you will have to go through some additional bioinformatics analysis to do the motif finding in those regions. But certainly, your resolution is much better than a few hundred base pair. And maybe you get down to the 30, 40 base pair. So there are special algorithms that are designed for that. So and we'll get through this sharing part in the later slides. OK, so what's the difference, the major difference between ChIPseq and ChIP technology? And first of all, of course, we have to say something good about the sequencing, right? And the first one is ChIPseq offers a higher resolution with advanced bioinformatics analysis we will talk about in the next lecture. And the less noise of ChIPseq, this is because of the no cross-hybridization, higher dynamic range, and everything we talk about why the sequencing is better than the arrays. And this is the interesting point, OK? If you're already falling to sleep and wake up here. So ChIPseq will offer you greater coverage. So what that means is once you got the ChIP, even though it's a whole genome tiling array, OK, and you design 25 base pair probe with the target on every 35 base pair resolution. But we know that only 48% of the human genome is non-repetitive. So in theory, you're targeting less than 50% of the entire human genome. So those repetitive regions are not going to be on the microarray because nobody in their right minds will design the probes to target on those. However, from the second lecture, I think it's second or third, I don't remember. But remember that mappability issue. We know that for human genome, that about 80% of the region will be mappable with 30 base pair rates and 89% with 70 base pair rates. This is the reason that there's a lot of human regions that are not like a huge repeats and all the way around. It's smaller repeats here, smaller repeats there, smaller repeats. Although they are not really on the probe design, but that will also be part of a rate, not the entire rate. So those rates, even though it contains the repetitive elements, but it's still mappable. So this is a major differences between the ChIPseq and the ChIP. It really have much greater coverage, and which is something we like. That make sense? Nobody even nodded their head. No? All right, we'll move on. We'll think about that. And the next point is simple. It's cheaper. So that's one of the major advantages. Actually, it's a lot cheaper, ChIP compared to ChIPseq at the current stage. And when we talk about short rates, so there's one issue that we talk about very early is they are too short. So is that OK? And we know that shorter rates is an issue for assembly, but it's not too much for ChIPseq. And so that part can be taken care of. OK? So much for the principles of a ChIPseq experiment. So basically, let me reiterate the approach. So the methodology itself is kind of straightforward, right? And then you do the cross-linking of the DNA. So the protein already binds here. You share the DNA into smaller pieces. You antibody the target on that protein, and then you sequence it. That's it, right? And so in the earlier stage, that there's a ChIP assay can do the same job, and now ChIPseq is doing much better. So that's a brief summary. So let me go through a few studies that is using the ChIPseq technology. And the first study is, of course, looking for transcription factor binding events. And this is probably the most straightforward application of ChIPseq. And the first ever ChIPseq paper is published in 2007. This is another seminal paper from Barbara Vo's group. Remember, when we talk about RNA sequencing experiment, the first paper also published from her group. And this is published in Science in 2007. And the transcription factors they were focusing on was neuron restrictive silencing factor, or NRSF. And you can read about their functionalities. But when you read the papers in greater detail, they did justify why they are picking this algorithm, this protein. Because this is the first paper, and they use ChIPseq. They want to characterize the performance of ChIPseq. And this protein has been really very well studied. And they also have around hundreds of validated target site, positive and negative sites. So they can later use that to characterize the performance of the assay itself. That's why one of the reasons they studied this. And the cell they use is a human T lymphoblast cell lines. And the platform, you can see this is the 25 base pair arrays. It was in a very early stage. Okay, there's some results overview, and if we look at the results today, and it's good experiment. Is that exciting or surprising? Probably not. But this is the first technology paper, so we have to give the credit for that. So you can see that this is the genomic locations. At this location, there is a ChIPseq protein. It's a protein binding site. And once you zoom in this area, what you look for here is you can see there's so many different short rays that is aligning along this region. And this is, so you can see this is the ChIPseq region, and they also have some type of control. By saying control, that means that they don't use antibody to target on anything. So it serves as negative sites, and you can see you really don't see anything around the control. And this is another biological duplication, and this is the control. So you can see very clear picture that there are pretty strong signals in the ChIP assay. So they did two conditions with biological replicates. One is this TF enriched sample. The other one is no immunoprecipitation control. And they did identify almost 2,000 loci in the human genome that with this protein binding, and in or near about 1,000 genes. So this is to the extent that this protein is functional. And the way that they did that is they want to see it's very early stage. There's no really any tool has been developed back in those days. And their definition is if they observe the 5 times more signals in the ChIP, the samples that in the control, and they will define that as an enriched region, which still makes sense if we look at from today. And more importantly, they did evaluate their approach. They found about 87% of sensitivity and 98% of specificity. And this is really based on known positive and negative binding sites of this protein in the neuronal, in this T cell lymphoblastoma cells. And you can see this is the ROC curve, and you can really look at it. This is a very high accuracy, and their ROC is their AUC, which is the area under the curve, reached 96%, and that is really very accurate prediction. And you can also see that they also did motif finding, right? And they found a better motif discovery using ChIPseq. So one thing they look at here is they identified these canonical motifs, which is 2 half sites, and there are maybe 2 base pair in the middle, and they say this is 11 base pair half site distance. That's the canonical binding motif. But they also found out there's many non-canonical binding sites as well. So you can see this one, the half sites are the same, but the distance is much greater. It's 17 base pair or something like that. And those are the ones that we probably will miss if we don't do this experiment. And you can also see the distribution, the number of sites that contain the canonical and non-canonical binding sites. So this is X here is the half size distance distribution, and Y is the number of sites that in those areas. And we will talk about how to use the different bioinformatics approach to identify these motifs in the next lecture. I think that is one of the major things you have to go through if you study ChIPseq experiment. Okay, and the second study is do the histone mapping. So the first one is the transcription factor, and the second one is histone mapping. So this is also published in 2007 from Keji Zhao's group at NIH. And so they did a very complete study, and this is published in Cell as a resource paper. The systems they use is CD4 positive T cells, and the protein they use is not one transcription factor, rather it's 20 histone lysine and arginine methylations. Okay, so the 20 different type of histone modifications, and histone variant H2AZ, and RNA polymerase II, and the insulator binding protein CTCF. So you can see this is, although it's the same year, same in 2007, and they did a much more complete mapping of the histone, different histone markers in these T cells. And the platform they use is also Illumina. At this stage, they are increased that to 36 base pair. And here are some of the figures I just paste and copy from their paper. So basically, this is for RNA polymerase II, different histone markers, okay, and the histone 2AZ. And so the point here is each figure centered at 0. 0 meaning it's the 0 distance from the transcription starting site of the gene. Okay, so the distance between the gene and any genomic loci. So you can see that we did observe very strong signal of RNA polymerase II around the transcription start site of the gene. This is not really surprising. But for the other study, for the other data, this is probably the first time that we have the opportunity to observe that. For example, we can see that this is what histone 3K4 trimethylation binding pattern should look like around the transcription starting site of the gene. And this is dimethylation, monomethylation, so you can see that there's a different histone markers, and there's a histone 3K36 markers, and for different markers, what it should look like in the genome-wide scale. And this is probably the first time that this has been really mapped. And they found that monomethylation of histone 3, K27, K-NET, these several histone markers, the monomethylation is linked to the gene activation, and trimethylation of these markers are linked to the gene repression. So those are the things that people still trying to figure out why that happened, but at least that is, with the ChIPseq technology, this is the first time people understand there's still different histone markers and their binding patterns in the active and the inactive the genes. So they also found the histone 2A associated with the functional regulatory elements, and also CTCF marks the boundaries of histone methylation domains. So if you're interested in all these things, and go ahead to read this paper. I'm not going to have the time to give you a very detailed introduction. The third application, it becomes really smart, and which is the enhancer identification. So keep in mind, the first application was transcription factor binding. The second one is histone markers. The third one is enhancer identifications. These enhancer identifications are something different. Okay, enhancers is really, really hard to find. We don't know where they are, and in the NA genome, because they are very distant from the transcription starting site. Sometimes it's hundreds, thousand base pair, or even millions of base pair away from the transcription starting site of the gene they are targeting. So sometimes it's even in a different chromosome. So they are not really easy to look for. And the second one is they are scattered among the vast non-coding portions of the genome. And those regions are traditionally defined as the junk genome. And they are evolutionarily conserved. That's a good news. So that can help us to identify the signals. But if only look for the evolution, you assume that this is conserved, so this is the enhancer. And I don't know how many false positives you will face. That is not a real way to do it, right? And also, it cannot predict when and where they are active as well. So it's hard to find. And people has been using P300 as a marker for the enhancers. And I'm not going to go through these figures, but this is the promoter, this is the enhancer. And what happens is you can see this P300 serves as one of the mediator proteins that brings those transcription factors in the promoter region and the proteins binds in the enhancer regions. So if you do and sometimes the P300 binding is in this way. So if you really target on P300, use your antibody, that is really helpful to identify the enhancer regions. And this is the way that people do it. There's 2 papers published from the same group, and both on Nature. One is in 2009, the other is 2010. So they were looking for the enhancer activities during the heart development. Okay, and I'm just showing some. The reason they are publishing in Nature, I'm pretty sure that this is not an easy experiment to do, right? I still don't know how they do that. This is mouse 11, embryonic day 11.5. All right, so at that stage, the embryo is still very, very tiny, very, very small. And then they were trying to look for tissue specificity. They target on different, very sub-small regions in the embryo, mouse embryo. It's already very tiny. And they did ChIP-seq on those. It's really not something that you do every day. It's just hard. But giving them credit, they published 2 papers in Nature, which is great. And one of the conclusion is enhancer regions are tissue-specific during embryonic heart development. So you can see this is the top one. You cannot read from the back. The top one is the forebrain, okay? And this is the midbrain, the middle one. And this is the limb, all right? And this is the conservation site. So you can see that in the forebrain part, and you are observing a very strong activation of these enhancer regions, but you don't see in other tissues. And for this particular enhancers, it happens in both forebrain and midbrain, and there's a tissue specificities around that. And this is probably, it's not really something surprising, but that's the first time people has a capability to look into those activities. And the next thing is they found enhancers are enriched at high conserved non-coding regions. So you can see that they look for the enhancer they identified, and they want to see the conservation score around those areas. Not really surprising at all. You can see that around the regions they identified, it's highly conserved. Okay? So you can somehow use the conservation to help you to see the confidence of your results. But you cannot really only use conservation to look for the enhancer markers, enhancer regions. That is too much false positives. And this is the enhancer evolution. This is very interesting results. And the enhancer conservation is tissue specific. So what this tells you is they found, I'm not going to go through the figures, but although you can read that pretty clear, so heart enhancers, the enhancers in heart, are much less conserved than the ones in forebrain, okay? And midbrain and limb lies in the middle, okay? People are still trying to figure out why, but it seems that is what's happening. So you can see this is for the mouse, and this is for different species, rabbit, so some of and this is fish, frog, and chicken. So the distance, more distance is and this is the divergence time, and 500 million years, and this is 50 million years, 100 million years, and you can see that if the evolution distance are smaller, and those are more the heart enhancers are enriched. But once you go to more divergence in terms of evolution, and most of the enhancers will be conserved in the forebrain. So those conservations are also tissue specific. That's something that helps us to understand about evolution. Okay? Okay, we talked about 3 different studies: transcription factors, Histone markers, and enhancers. And next one is DNA methylation. And this is, to be fair, shouldn't be called ChIP-seq, right? And because ChIP, the first ChIP stands for chromatin immunoprecipitation. But here you are using different ways to target on the samples, which we'll talk about that in the last lecture, and which more focus on epigenetics. But there's more than one type of methods. The first one is immunoprecipitation-based assay. This is a MIDIP-seq or ChIPseq. I'm not talking about that today. And there's also methylation-sensitive or insensitive restriction enzyme-based assay. So you use those restriction enzyme to treat the DNA. Sometimes if there is a methylation, it's protective or just specific cut in those regions. And then you do the sequencing, you're able to find out something. And the most hyper method is called bisulfite sequencing. And it requires but there's a nice thing about bisulfite sequencing is it gives you the one base pair resolution in terms of where the methylation occurred. But the problem for that is it requires full genome sequencing for the control and the bisulfite treated samples. So a lot of resources are required. And also, but sometimes it can work on restricted regions and reduce the representation of bisulfite sequencing. So this really doesn't say too much because it's too abstract. But once we get to the epigenetic lectures, I'm going to give a little bit more detail on those assays. And these methods differ in their capabilities to detect differentially methylated regions between pairs of samples. And there's a very nice review paper about the comparisons between different approaches. And the next study is our study, and it's in collaboration with Jeremy Sanford at University of California, Santa Cruz. And it's a study of the mapping of RNA binding proteins. So here we are not really doing ChIPseq. It's called the ChIPseq. The ChIP stands for cross-linking immunoprecipitation follow the sequencing. Okay? So what happens is very similar to ChIPseq. Rather, it's study the protein-RNA interaction, not the protein-DNA interaction. The experiment itself is just instead of using formaldehyde to do the cross-linking, we use the UV light to introduce those protein-DNA interactions. So there's a small difference between this and using formaldehyde. We talk about using formaldehyde, you will introduce the protein-protein interaction as well. But use UV light, it won't introduce the protein-protein interaction. It's only if the protein-RNA or protein-DNA interactions. And after you're doing the cross-linking, and you do the regular steps, and then you use antibody to target on the RNA binding protein we're interested in. In this case, we're interested in a protein called SF2, and then you further do the sequencing. As you can see, this is the study was done in 2007, and at that time there's no even Illumina, so it was a 454. And as you can see from today, that at that time, it only provides about 400,000 reads of 150 to 200 base pair. We know today the sequencers are much more capable, but that's the stage that we were at in early 2007. And the analysis itself includes several different steps, so including the removing the linkers and the sequence filters, and to do the sequence alignment, and that alignment was black. Right? We don't have any fast aligners available back in those days. So we used black, and we should burn several of our machines, but that was OK. And then we do the annotation and binding site discovery and functional analysis and the gene ontology. So you can see that all these different analyses or different components that has been well established. But our goal here is to use those information and trying to understand something about this important RNA binding protein. By the way, this was the first RNA binding protein transcriptome-wide mapping ever available. And there's some of the results, just very briefly introduction. So you can see that the annotation of SR protein binding sites, so that we found about 75%, 73% of the sites we identified are in the gene region, okay? Which makes perfect sense. That tells us the experiment really did well. And about 1/4 of those regions are out of the genes. And for the ones that within the genes that we can see that it's a lot of them, or 3/4 of those are associated with exons, and others are not. And this is the motifs that we identified, and we can see that this motifs gives us the prediction power, about 80% of prediction power in the ROC curve. And we also see some of the binding sites and are targeting the splicing junctions, meaning that these proteins are binds on the mature messenger RNAs, although we also see some of the evidence of the precursor mRNAs. And these are the gene ontology analysis. Actually, one of the interesting, you can read the paper, one of the interesting fact is this is RNA binding protein, and the categories that they're targeting is also RNA binding protein. So it's kind of a cascade type of regulatory network. Okay? And so that is the study of RNA binding protein using ChIPseq assay. And so that is really goes into the alternative splicing field. So I didn't show you the result, but you can read the papers if you want. There are plenty of disease association related to regions we identified, and they are very much enriched in the alternative splicing pathways. Okay? And the next study I want to talk about a little bit is to study microRNA. And people may think, you're crazy, it's not possible. But this study is done in Barbara Darnell's group at Rockefeller, and he invented the ChIP assay. So we know that this is a microRNA function. So you can see this is a messenger RNA, and this is the red part is the microRNA. And then we know that when the microRNA is working, and it will bind on the 3' UTR of the gene, and at the same time, they recruit a protein complex called the risk protein complex, and then do the killing part, right? This risk will kill the messenger RNA or stop the translation. But this has been, the people start to take advantage of that. So this is the crystal structure of that. So you can see this is the blue one is the messenger RNA, the red one is the microRNA, and this is the risk protein complex, which one of the proteins is AGO. And this is the protein become a target in the ChIPseq assay, and people start to really design the antibodies to target on AGA. So they use the ChIPseq assay. They first use UV light and to introduce this protein RNA interaction, and then use antibody to target on this protein. Okay? In theory, after doing this, you get all the microRNA targets in the cell into your pool of sequencing. And then you further do the sequencing of that. Does that make sense? And again, this was published last year in Nature. But since this is an assay that nobody has ever done it before, and they really did a very careful analysis on the reproducibility, and with the 3 different brains, they saw very excellent reproducibility of the results they identified. And they also look for the target distribution of this microRNA, because all the regions they put down, the sequence, are at the moment the microRNA targets. So what they see is, so you can see this one, 25% of those. I mean, this is for a long time that we think that microRNA is working the 3' UTR, and people always say that, but there's no way we know whether it's right or not. And before this study, there's no experimental way to look for the targets of the microRNA. Now this is the first time we know that a little better. The first, you see, about 25% of the microRNA targets are actually targeting in the coding region. All right, this is I'm not sure that is surprising or not, but that's something that's good to know. 32% is in the 3' UTR, and 12% is in the intron region, and 8 additional % is in the 3' UTR extension region, so the region that is pretty close to that. Okay, and they also find about 5% are in the non-coding RNA regions, meaning that those non-coding RNAs can potentially be the microRNA targets as well. Okay. Overall, this is the target distribution that is the first time we have ever seen. And this is they also did a pretty careful bioinformatics analysis to follow this up. And so you can see this, because what you get out of this assay is not a specific target on certain microRNA. It's all the microRNA targets, and it's a combination of many different microRNAs. So the region you identified, you know at the moment which microRNA, you know at the moment it is the microRNA targets, but you don't know which microRNA was targeting that. Okay, so they do need to know to get a very deep bioinformatics analysis to further flesh out that which microRNA was targeting. I'm going to stop here today, and the homework is due today, by the way. So if you haven't sent it to me yet, and please send me through email, and I can also receive hard copies if you want. And for the students registered class, please stay. We need to distribute the quiz. We don't have new quiz today, but we need to distribute that.